Okay, so drawing the chemical was a little buggy, so I'm going to take another stab at it. All right, so the chemical version of this is we're just going to break down what each of these parts actually is. So this phosphate group is an oxygen bonded to a phosphorus, which is double bonded on either side to an oxygen and bonded to another oxygen like this. And this oxygen bonds to the ribose, right? So this here, this is this little guy right here. This is the phosphate group. Just this little P over here. So there's kind of like a line going between these two things. And now we're going to draw the ribose, what the ribose looks like. And I could just as easily draw a deoxyribose, but we're going to go with ribose here. And so here we're going to draw a carbon and a hydrogen. And that's actually two hydrogens. And that's bonded down to another carbon, which is bonded to an oxygen, another carbon, which is bonded to a hydrogen. Don't pay too much attention to all this part. This is kind of just filling everything in. There's a carbon here, and there's a carbon here, and it forms a nice little ring. Okay, this is the ribose. And there's hydrogen here, there's a hydrogen here. Here there's an OH, and this is kind of important, this part down here. We're going to talk about this in a second. There's an OH here. This is the part that makes this ribose. If this were deoxyribose, there would not be an OH here. There would just be an H. Okay, so this is the thing that makes it a ribose. And over here, it goes off to the base. And I'm not going to draw the base in because there's 4 different bases, and I could draw them each differently. But we're not going to talk about that part right now. The most important thing to know is that they're all chemically different, and they all sort of base pair with each other differently. And that's what gives them their special pairing properties and what makes them unique. So here we have the phosphate group. The base is here, and there's this little hanging OH group here. Now, this OH can react with another phosphate, and then that can give you instead of an OH, that can give you an O going to another phosphate group, right? And now you're starting to see the chain repeat itself. Okay? And then this O here, this goes to another carbon, and then, down here, and I'm running out of space, but then over here, this could be, another ribose. So this is the phosphate, this is the big P, this is the ribose right here, this is another phosphate, and then down here would be another ribose, and coming off the side here is a base. Now there's one single little detail besides the fact that these phosphates bond to this little OH here and replace it, which is going to be important later, and that is that this carbon is named the 5' carbon. And the reason it's called the 5' carbon is because if for some reason you count around from this carbon, and this is the 1' carbon, this is the 2' carbon, this is the 3' carbon, and this is the 4' carbon. And you'll notice that on one side of the ribose, the 5' side of the ribose is bonded to one phosphate and is bonded to another, presumably another ribose up here. And down here, the 3' side is also bonded to a phosphate and is also bound to a ribose. But that it's not symmetrical, right? So see how this carbon is kind of hanging off of the big group here, and this part is not hanging off of the big group here. You can't sort of, if I put a line through the middle here, you can't flip this and have it look the same. And what this means is that DNA has a directionality to it. All right? Directionality. That's not even readable. It has directionality to it. And this side is called the 5. That's a P. Let's not make a P. Whoa, that's crazy. Well, okay, so that's not going to work. This is called the 5' side, and this is called the 3' side. And when you draw any piece of DNA, there's always a 5' direction and a 3' direction. And this is going to be oh so important later when we talk about polymerase, because polymerase only works, for whatever reason, it only works in one direction. So we'll talk about that again. Anyway, so that's just all the pieces of the puzzle together, and this little detail about 5' and 3'. And you can watch this over and over again until you're blue in the face, but I'm not trying to make you memorize what these structures are. I had trouble drawing it out myself. The key thing to remember is that these phosphate groups are bound to the ribose. There's a base hanging off of each one, and that there's a directionality to the whole thing. And beyond that, I won't be expecting you to know a whole lot more, and you'll see how the details come in and fit in as we move on to polymerase. Okay, so that's enough repeating myself. That is all for this video.